All right, we'll go ahead and get started. Thank you all for coming, everyone. We're gonna move forward. We're gonna, introduce Dr. Efimov. Thank you for joining us today for the September 2023 monthly seminar on physical genomics. My name is James Peterson, and I'm the Senior Director of the Center for Physical Genomics and Engineering here at Northwestern. The CPGE is a transdisciplinary research center that's focused on studying the structure and function of the genome through the convergence of super resolution imaging and nanosensing technology, modeling, computational genomics, biology, and artificial intelligence, with the mission to develop new strategies for the treatment of disease and the reversible engineering of living systems, and also to train the next generation of multidisciplinary scientists. You can learn more about our research and training programs at physicalgenomics.northwestern.edu, or follow us on YouTube, X, i.e. Twitter, or Instagram. We are very excited to welcome, as our speaker today, Dr. Igor Efimov. Dr. Efimov is the director of the Cardiovascular Engineering Laboratory here at Northwestern, an NIH-funded cardiovascular research institute and engineering laboratory that focuses on the physical mechanisms of cardiovascular disease and on developing novel therapies for heart diseases, with an emphasis on heart rhythm disorders. Dr. Efimov, thank you so much for sharing your work with us today. For our audience, there will be time for questions at the conclusions of the talk, or you can type them into the Zoom chat at any time, and we will address them during the Q&A after the talk. At this point, Dr. Efimov, if you would like to share your screen, we can begin your presentation.
Yeah, I'm already sharing my screen.
Oh, perfect. Yes. I thought that was ours. That's the Center for Physical Genomics and Engineering. Thank you so much.
Yeah. Thank you for this opportunity. I'm really happy to present my reason why I'm interested in physical genomics. And I have to admit that there are a number of reasons why we moved to Northwestern last year. And one of them is actually my interest in physical genomics, which has been slowly growing over the last 5 years. And I would like to show you our journey, why I think it's important to answer many questions which we have. So these are my disclosures. Really, I will not sell you anything today, so it will be easy. So let me start with actually normal physiology, but I think it will open some line of reasoning why I think this is important in the disease as well. In 1906, Sunao Tawara, a Japanese postdoc who was working in Germany at the time, so he discovered what we now call AV node, atrioventricular node. And he published, this is his publication right there in 1906, a book which was exquisitely illustrated. He depicted all these histological structures himself. They're really beautiful. You can see here some cross sections of AV node. And basically what he observed was that on the intersection, on the interface between atrial septum, which is shown here in yellow, and ventricular septum shown here below in yellow, there is a structure made of connective tissue, which is connecting atria and ventricles. And this structure, of course, does not contain myocytes, except for one region where there is a bundle starting in the atria, and this is AV node, and then goes straight through this whole connective tissue structure as a His bundle, and then it splits into two branches, left and right bundle branches. So he observed that even though these are myocytes and the AV node, but they are clearly very, very different from the rest of myocardium. They have different shape, they're very small, they have very little evidence of contractile proteins, almost no sarcomeres. They have very few mitochondria, so basically metabolism is very different. And as we learned later, they have very different electrophysiology. So the structure is peculiar, very different from the rest of the myocardium. A year later, in 1907, Keith and Fleck in England, following the work of Tawara, they actually described it in their paper. They discovered similar structure, similarly looking structure, at the interface between superior vena cava and right atrium, basically on top of the same atrial structure. Here's the cross-section, and this is histological slide. Same story, very different cells, very small, no metabolism is different, mechanics is very underdeveloped. And then again, as we learned later, electrophysiology is also very different. And also it's encapsulated in connective tissue. So we'll skip a century of research, and basically what we know now is that there is essentially a structure which we can call fibrous skeleton of the heart, which is essentially boundary made of connective tissue between the atria and ventricles. There are also structures which attach leaflets of major valves, and they are shown here. And what is interesting, all these fibrous skeleton structures are adjacent to myocardium, which has very different properties compared to normal working myocardium. They all possess properties of pacemaking cells at different frequency. They have very underdeveloped mitochondrial system and metabolism in general. They also have very underdeveloped mechanical function, almost no sarcomeres, very few sarcomeres. So they are very different, these structures, in other words. So what exactly makes them different? I think this is what physical genomics can help us, because I think it shows that there is a link between mechanics, because these cells really do not possess a significant mechanical function because they are embedded in connective tissue, and their nuclear architecture, their transcriptome. So let me first show you very quickly a project which we did over the last few years, in which we ask ourselves the question: can we look at how many pacemakers we can detect in normal rat and then human heart? To achieve this goal, a student in the lab, Jacqueline Brennan, she developed optical imaging technique where you basically excise right atrium of the rat's heart, shown here on the right. Then you stain it with voltage-sensitive dye. You are recording action potentials from about 7,000 sites. And then you can detect each action potential shown here in each site. And then using signal processing, you can determine what is the site of origin of the heartbeat, where does it come from, which is essentially a representation of pacemaker in the heart. When she did that, she also perfused the heart with isoproterenol to stimulate sympathetic system and accelerate heart rate, or with acetylcholine, in which case, of course, heart rate goes down. And in rat heart, normal innervated in vivo heart gives you a heart rate of about 400 beats per minute. You can bump it up with isoproterenol close to 500, and with acetylcholine, you can go down to 100. So basically, the entire range in the healthy rat heart is between 100 and 500 beats per minute, approximately. So when she actually looked at location of pacemaker, and this graph shows all the locations from all experiments. She did 8 experiments with all the concentrations of isoproterenol or acetylcholine. As you can see here, each of these circles is the location of pacemaker in different hearts and under different conditions. So red circles show location for stimulation with different concentration of isoproterenol, blue acetylcholine, and black unstimulated, basically no autonomic involvement. So you can see, even without doing any statistical analysis, but we did it, of course, that there are 2 clusters. Here's on the right, there is a probability distribution. There are 2 clusters, one cluster on top and another cluster at the bottom. So this vertical axis is between superior and inferior vena cava. So if you project that these locations on the depiction of the heart, you see it below. So top structure, basically what is traditionally known as SA node or sinoatrial node, which was discovered by Keefe and Flagg. And the bottom structure, we basically called inferior sinus node. So now we claim that there are 2 sinus nodes in the mammalian heart, superior and inferior sinus node. Superior sinus node is responsible for faster frequencies. That's why it's dominant, and most of the time we see only that particular structure. That's why in your textbooks, when you study biology, only one SA node is shown. The other one, it only has a chance to work when you withdraw sympathetic stimulation, and then parasympathetic system will kick in and basically will exhibit behavior of the inferior sinus node. So we proved it in a number of experiments. For example, if you cut atrial preparation with scalpel, you will see behavior of both sinus nodes, because now a slow sinus node is not inhibited by fast sinus node. If you apply acetylcholine at high concentration, you completely inhibit superior sinus node, meaning that's why inferior can kick in. And if you wash out acetylcholine and apply isoproterenol, you accelerate both pacemakers, and you can see that superior is faster, and in an intact heart, it would have been dominant. So we did whole tissue RNA sequencing, and tissue was isolated from superior sinus node, inferior sinus node, right atrium, left atrium. We also had ventricular tissues, but I will not focus here on those. And if you look at heat maps on the right, you can see that superior and inferior sinus node is the two vertical columns here on the left. They clearly share a pattern with one another, but slightly have different difference of levels of expression. So these are cardiac ion channels, cardiac receptors, neural proteins, transcription factors. And you can see they are also different from both right and left atria. So they have quite different transcriptome, but they do have similarities between the two sinus nodes. And atria also are somewhat similar between each other. I don't have time to go over all these details, but they are quite interesting and they are published. You can look up the paper. Here is just a close-up, just looking at cardiac ion channels. Again, you can see here, for example, on top, these are specific ion channels. There are potassium, calcium channels, gap junctions. This is connexin 43, for example. So you can see levels of expression in the atria on the right versus sinus nodes on the left, very different. So then we did the same experiment in human heart. In the human heart is a donor hearts, which we again isolated like in the rat, and also demonstrated that yes, in human heart, like in the rat, we have superior sinus node, which is normal sinus node, and inferior sinus node. And again, we looked at transcriptome from those structures again, as in the rat heart. So we clearly see that superior and inferior sinus node have differences versus atrial tissue, but similar between each other. Interestingly, when you subject rat heart to transverse aortic constriction and you put essentially suture around aorta and you introduce hypertrophic cardiomyopathy and heart failure subsequently, so here is a failing heart versus normal heart of the same age, you can see this dramatic enlargement of the heart. So when we looked at sinus node in these failing hearts, we found something very interesting. This in panel C, you can see that in 4 hearts, we studied 6 hearts here. In 4 hearts, all conditions of stimulation, sympathetic or parasympathetic, did not move the pacemaker. It remained in the superior location. In 2 remaining hearts, they all remained in the inferior location. So basically, mechanical stimuli which are produced by aortic constriction and enlargement of the ventricle result in remodeling of atrial myocardium. And as a result of that, there is a change. There is suppression of one of the pacemakers in the heart. So we did subsequent experiment in which we took 4 hearts. We first determined which pacemaker is present in these failing hearts. In these 4 hearts, all 4 hearts, only superior pacemaker was present and inferior was not present. So again, we did RNA sequencing. So I don't have time to go over details. It's very interesting remodeling process. But when we compared what happened in the superior sinus node, which I remind was preserved versus control hearts, we found some remodeling, but not a lot. So all these green colors represent downregulation or upregulation in ion channels, cardiac receptors, neural proteins, or transcription factors. But if you, again, this pacemaker in these hearts was preserved. But in the inferior pacemaker, which actually was suppressed by heart failure, by mechanical dysfunction, we found whole host of proteins of transcripts which were either up or down regulated, involving all major ion channels, sodium, potassium, as you can see here, calcium. TBX5 and TBX3 were remodeled with the major transcription factors in the atria. And interestingly, a lot of actually structural proteins. For example, here, ankyrin 1, 2, 3, these are structural proteins which assemble membrane proteins in multi-protein complexes. So this perhaps shows that this mechanical dysregulation is one of the reasons to produce dysfunction of sinus node. And previous work done by Mark Anderson and Peter Moller from Iowa showed that mutations or knockdown of, for example, ankyrin 2 results in sinus node dysfunction. So we were not surprised to find these targets. So there's clearly a link between change in transcriptome, rendering pacemakers active or inactive, which depends on mechanical dysfunction. So now I'm going to switch completely to a new topic. So we study in our lab for many years human hearts. So I started this when I was at WashU in St. Louis about 15 years ago, and basically this is procurement of male and female hearts, both failing and non-failing. We did also this at George Washington University, and now we started this in Chicago. So what we do with that, so we receive hearts, which are live hearts, arrested cardioplegically. We can perfuse them, conduct functional physiological experiments using electrophysiology, mechanics, metabolic function assessment. We also create slices from these tissues and can study again all these functions, mechanics, metabolism, and electrophysiology in cultured conditions for several days, up to 1 week. Or we do various molecular studies. I don't have time to go over all these techniques. I will talk only about our epigenetic and transcriptomic studies. So early studies which we did a number of years ago at WashU, this is one of such papers. We showed that if you take samples from non-failing hearts shown here in black versus 3 types of cardiomyopathy, yellow is ischemic heart disease, essentially patients with myocardial infarction. Magenta is dilated cardiomyopathy, and red is septic cardiomyopathy. So you can see that ischemic and non-ischemic or dilated cardiomyopathy are at the end stage, and that's the type of samples we can only get. They do not distinguish from one group to another. They really, in the principal component analysis, are indistinguishable based on bulk tissue RNA sequencing. However, septic cardiomyopathy is very, very different. So it differs remarkably and has a very large number of genes which are differentially expressed. So this affects very strongly mitochondrial function. So here are many targets which are downregulated. There is essentially widespread downregulation of mitochondrial genes in septic cardiomyopathy. And also importantly, there is a dramatic downregulation, as you can see here, in electrical and mechanical systems. So for example, here is a calcium system. So SERCA, phospholamban, ryanodine receptor, all downregulated. Also, if you look at junctions, cell junctions, plakophilin, desmoplakin, and also connexin 43 here, all downregulated. Also, many genes, as you can see here, are downregulated in sarcomeres. So essentially, electrical, mechanical function, mitochondrial function are all affected by heart failure. However, again, in a larger study, which we conducted later with much larger number of samples between Utah and the Innova Hospital in Virginia, so we showed that basically, again, ischemic and non-ischemic cardiomyopathy cannot be distinguished, and there is even a theory emerged that perhaps at the end stage of these different etiologies of heart failure, they converge and cannot be distinguished from one another. But I think it's really not about mechanism, but rather about limitations of RNA sequencing as such. So subsequently, we did a number of studies on epigenetics, on acetylation in particular, or methylation. And I'll just highlight one of such studies, in which case we looked at acetylation in 20 hearts with severe aortic stenosis. These are septal biopsies from patients with severe aortic stenosis during valve repair surgery versus healthy controls. And we found that basically there is approximately 6,000 regions which are hyperacetylated and 6,000 which are hypoacetylated. So it's not very informative, of course, it's a lot of targets, but and they distinguish quite nicely between aortic stenosis versus control. But bioinformatic analysis showed that actually this very large number of differentially acetylated regions correspond to only 52 genetic loci, which correspond to different transcription factors. And in reality, only about 10 of them are really most implicated. So you can see some of them are very well known, like CREB, for example, 3 and 5. And perhaps this could be interesting therapeutic targets. But again, it's not enough insight to develop a strategy for treatment of this disease. Therefore, we several years ago started and published finally this year work on using KH technology, which is CAP analysis of gene expression with a group from Japan. And we developed an atlas of transcribed human cardiac promoters and enhancers, and we compared it in different disease states. So basically what we found, as was highlighted in this nice editorial in the January issue of Nature Cardiovascular Research by Vincent Christoffels from Amsterdam, that we found basically that numerous genes which are implicated in heart failure or other diseases of the heart have multiple promoters and enhancers. And what is interesting, the healthy state versus disease state, they select different promoters for transcription of the RNA and then ultimately for production of proteins, which results very often in different isoforms. So first of all, very briefly, what KH technology is about. So normally, if you run RNA sequencing or microarray, so you basically focus primarily on protein coding region, and you more or less ignore the ends, and you basically really don't resolve very well promoters or enhancers, especially enhancers. KH also works with RNA, but actually it focuses only on regulatory elements and only sequences the promoters and enhancers regions, allows identification with very, very high precision down to single molecule. So essentially, after you isolated these regions, cap regions of RNA, and then you sequence them, we map it back to sequence of DNA, and then such Manhattan plots are plotted showing height of the peak corresponds to activity of a particular regulatory element, such as, again, is promoter or enhancer. So in our study, we used 21 donor hearts and 10 failing hearts shown here, almost equally spread between men and women, and also covering age from 26 years of age to about 75 years of age. So first, big picture, we identified very large number of transcribed regulatory elements, about 55,000 of them. So out of which we have a comprehensive library of bidirectional enhancers, 17,000 plus promoters, and almost 15,000 enhancers. So we conducted exhaustive validation versus already published data. Also, what is nice, because it's a quantitative method without involving any amplification, so it's much less noisy compared to RNA sequencing. So we can compare, for example, in this case, this is bidirectional enhancer for this gene KLF9, which is a very important gene in heart failure. So we can see that, for example, comparing failing and healthy hearts, there is a big difference in expression or activity of this enhancer. There is a big difference between atria and ventricles as well, and between left and right heart. So again, we validated it against a number of databases. So here is, for example, data comparison with ChIP-seq, ATAC-seq, and DNA-seq for the most reputable published public databases. So our data corresponds nicely, but presents a much more accurate representation of regulatory elements. So again, we did this heat map comparison for enhancers. Again, we identified many more new targets, and also we compare very well with already known ones. So first, big picture view on comparison between failing and healthy hearts without distinguishing ischemic and non-ischemic cardiomyopathy shows that essentially we have healthy atrial cluster and healthy ventricular cluster, which separate very nicely. But in failure, it was very interesting that failing ventricles collapse to a much smaller distribution, partially overlapping with healthy ventricles, whereas failing atria, shown here in orange color, split into two groups. One group still clusters with a normal healthy atria, while the other one actually goes into ventricular phenotype. And I will focus on it in a minute. We also found that there are very interesting differences between atria and ventricles with respect to mitochondrial remodeling, with respect to also defense remodeling. So then we looked at ischemic versus non-ischemic cardiomyopathy. Specifically, I remind you that in the early RNA sequencing studies, we could not distinguish these two for whatever reason. Maybe number of samples was not sufficient, but I think it's still a limitation of RNA sequencing technique. With KH, we have identified very large numbers, you can see here, specifically active promoters and enhancers in non-ischemic cardiomyopathy. There are 323, or ischemic cardiomyopathy, 255. And again, big picture is that non-ischemic cardiomyopathy is distinguished by remodeling in the immune response, defense response, cytokine-mediated signaling, for example, pathways. Whereas ischemic cardiomyopathy, as you look here, it's primarily related to metabolic downregulation. So there's a big difference really between ischemic and non-ischemic cardiomyopathy. Now let's go back to the story I mentioned about atrial cells now actually becoming ventricular. We called it ventricularization following an early study done in 2005 when a group from Germany identified that patients with chronic atrial fibrillation, if you run RNA gene array, what was available in 2005, they show essentially a pattern of expression similar to ventricular cells rather than atrial cells. They called it ventricularization. So now what we found was the same observation is actually applied to hearts with heart failure. So what you see here is a digital cytometry, even though this was not a single cell cage, which we're now working on, but still using decomposition methods, we can essentially identify different cell types within bulk tissue cage analysis. And you can see here different cell types: adipose, atrial myocytes, ventricular myocytes, neuronal, etc. So what you see here on the left, healthy atria, of course, have mostly these brown cells, which are atrial myocytes. On the right, ventricular cells have mostly pink cells, which are ventricular myocytes, and they preserved healthy and failing ventricles still have majority of ventricular myocytes. But what was really peculiar, in the failing atria, we found that small number of tissues have still atrial cells as majority, but 2/3 of samples actually had ventricular-like cells. So basically, there is ventricularization, remodeling of cells into ventricular phenotype caused by heart failure itself. So this is further quantitative validation of what I just said. Again, these are failing cells which colocalize with healthy atria, and these are failing atrial cells which colocalize with ventricular samples. So the ventricularization basically happened in 2/3 of the samples. And here are some markers which are, you can see, downregulate or upregulate in ventricular compartment, and they are specific genes. So then we started looking into actually how promoters and enhancers behave as a state of, for example, heart failure. So we identified there are actually hundreds of genes which switch promoter activity between healthy and failing heart. So for example, here's one of such genes, palladium. This is a cytoskeleton associated protein which is implicated also in heart failure. There are mutations associated with hypertrophic cardiomyopathy. It has two promoters as shown here. And you can see that, for example, this promoter on the right is very active in the healthy heart, and expression of that promoter is downregulated in failure. But the other promoter on the left, you can see now switched and actually active in the failing heart. So then we also identified hundreds of genes which have a specific etiology-specific activity. For example, in this case, this particular gene, angiopoietin-like one, which is a VGF family gene, is active specifically in non-ischemic cardiomyopathy compared to ischemic cardiomyopathy. So there is essential specificity to etiology of heart failure, to which promoter will have most activity during transcription. Then we looked at a recent paper published in Circulation, which was a large clinical consortium, which identified this list of genes for which there is a strong clinical evidence exists that these genes are implicated in pathogenesis of cardiomyopathy, both hypertrophic and ischemic cardiomyopathy. You probably recognize many of these genes. So there is alpha-actin, for example, desmin, desmoplakin, lamin, which physical genomics community knows really well, myosin heavy chain 7, etc. And we looked at how many promoters and enhancers do these genes have, and you can see here that many of so each circle corresponds to basically transcribed regulatory element, could be promoter or enhancer, and you can see there's a very large number of such elements. For example, in alpha-actin, in desmin. Some genes, unfortunately, don't have as many. For example, I was disappointed to see sodium channel only has one, or myosin heavy chain has also only one, but everyone else has multiple. And also you can see, based on the color of the circles, that some of these are actually downregulated in ischemic or non-ischemic cardiomyopathy here, for example, versus upregulated here. So there is a switch in heart failure for desmin, for instance, from one dominant to less dominant promoter. So we also looked at comparing to GWAS studies published in the literature, what is the location of known mutations associated with different cardiovascular diseases versus our library of promoters and enhancers. And we found that these are different diseases, as you can see here, for example, atrial heart septal defect, or familial hypertrophic cardiomyopathy, or resting heart rate, or atrial fibrillation, or cardiomyopathy in general. So we found basically that 40% of mutations identified by many different groups are located not in protein coding region. This green part is protein coding region, and this is not protein coding region. And also, depending on the disease, up to 20% of them are located actually in the site of promoter or enhancer. And also here on the right, you can see yellow color corresponds to promoters and pink to enhancers. So it's a very large number of mutations associated with cardiac diseases are actually located in non-coding regions and specifically in regulatory elements. Now let's switch to second part of my presentation about sex dimorphism of promoters and enhancers. So cardiac disease is very much sex dimorphic. There are many diseases which are specific to women or to men. Of course, there is also differences in physiology. So male heart is approximately 300 grams versus female heart about 250 grams. Male heart pumps 5 liters per minute approximately. I'm sorry, volume is 5 liters. Females have about 4 liters of blood. Heart rate is higher in females compared to males. And over a lifetime, women produce many more heartbeats compared to men, of course, in part due to a longer lifespan. There are, of course, differences also in hormones between men and women. So one of the most striking differences in cardiac electrophysiology is the difference in QT interval. So women tend to have longer QT interval. There is also morphological difference in J point and also T wave rate on the upswing here, which corresponds to differences in expression of different ion channels. There is also mortality from cardiovascular disease in women is much lower until age of approximately 80 years of age when they become equal. So what would be a possible explanation to these differences, or where do we go with this? So there are a number of diseases shown here which are specific, have higher prevalence in males on the left versus females on the right. For example, ventricular tachycardia, for still unknown reason, is much higher likelihood to be in women in the right ventricle and right ventricular outflow tract, while much higher in the left ventricular outflow tract in male heart. Brugada syndrome is preferentially male disease, while long QT syndrome is preferentially female disease. Similar Tarsat-de-Pont arrhythmia, ventricular tachycardia, is also found in women more often, while CPVT is more often in males. This is one example on the right. So this is a study published about 20 years ago where a Japanese group reported 748 cases of ventricular tachycardia, which they located as the focal source of arrhythmia, and they ablated it. And they found that right ventricular origin of this particular ventricular tachycardia is much more prevalent in women, so 142 versus 71 in males. While left ventricular tachycardia is actually, especially in left ventricular tachycardia, this is prevalent in males, as shown from this value. So we have a study on the way. Right now, it's not yet published, but reprint is available, in which we compared basically activity of promoters and enhancers in male versus female heart. And we also correlated it versus disease state, different cardiomyopathy, and also age. So again, what we did essentially, we took samples from male and female hearts. So we identified with KH regulatory elements, we sequenced them and identified their activity. And then we compared it in a subset of samples to RNA sequencing, which allowed us to compare basically different isoforms, which result from different usage of regulatory elements. So first, a big picture view, when we conducted principal component analysis on all samples, all atrial, ventricular, male, female, all ages, healthy and disease, found that essentially most variants almost 50% in principal component 1, comes, as you can see, between circles and triangles, which is basically between atria and ventricles. Male, female principal component analysis are not distinguishable, or disease state. Principal component 2, shown here in two different views. So this particular view is essentially healthy versus failing state. You can see that on the left, failing group clusters here, while healthy on the right primarily, or as ejection fraction. So again, you can see the intensity of the color represents ejection fraction, with dark color being very low ejection fraction. You can also see that low ejection fraction clusters on the right, on the left, I'm sorry, while high ejection fraction tends to be on the right. So basically, 10% of the variance comes from a disease state. And finally, the principal component 3, we looked at it, and you can immediately see red color on the left, blue color on the right, with high significance. Male and female heart status, sex status basically is the reason for variance, which accounts for about 5% of total variance across all the samples. So it's a very important difference, about 5% of variance. First, we looked at whether these differential activity of promoters and enhancers happen only in sex chromosomes or across the genome. And you can see, of course, here, this is for healthy hearts. Blue color represents male activity and red represents female activity. So you can see activity goes across all chromosomes, not only sex chromosomes. Same is preserved in failing hearts as well. So then we looked at what is essentially the difference between hearts. So here is first plot showing difference in the atria versus in the ventricles in the healthy subset, a healthy cohort. We had, like I mentioned before, 21 healthy hearts. And we identified that in the atria, there are 364 genes for which there is a differential activity between male and female hearts, and 558 genes in the ventricles, again, difference between male and female hearts. And you can see which systems are most prominently different. These are cytokine stimuli, a cellular response to stimulus, contractile fiber, basically mechanics, or immunity. So immunity, especially in the atria, is strongest in the female atria. This is, again, healthy hearts. Now let's look some examples. So this is a very important gene, HOPX, which is a homeobox gene, and it promotes cardiogenesis by activating BMP or inhibiting Wnt. So here's a comparison between KH data versus RNA sequencing data. So on the left, you can see that this particular gene has essentially two different active promoters, and you can see they're actually, based on intensity, are preferentially male, both in atria and ventricles, and much less active in female heart. And this corresponds to actually RNA sequencing as well. So there are two isoforms triggered by, started by these two promoters, and they are also much higher expression levels in the male atria and ventricles compared to female atria and ventricles. In the ventricles of a female heart, it's almost actually absent. Now we're moving on to sex difference in the failing heart. So you can see here we identify even more targets. Atria of failing heart have 636 differentially expressed promoters and enhancers in the atria, and 258 in the ventricles. So again, you can see here what particular structures are actually affected. So this is developmental processes, cardiac biology, homeostatic processes, and again, response to stimulus. For example, again, response to stimulus now, remember in the healthy heart, it was most prominent, most active in female atria. Now it moved actually to male ventricles. So here is an example. So this is a protein convertase subtyrosine, caxin type 1, which was implicated, for example, in diabetes. So you can see here that in the healthy heart, KH identified that there is one promoter which is active both in male and female atria, but absent in male and female ventricles, while in the failing heart, it's upregulated actually in male atria, but completely ablated, gone in female atria. So it's a very strong sex dimorphic response to heart failure. We looked at our favorite genes, transcription factors, and which transcription factors are most sex dimorphic. So they are shown here. and again, I already mentioned CREM, CREB, I'm sorry, in the previous, so CREM is another family. I'll talk on the next slide shown here on top, but I also will stop at TBX5. So TBX5 is invariably identified by many studies as one of the leading candidates for pathogenesis of atrial fibrillation. So it's an atrial gene preferentially expressed in the atria. Mutations in this gene or change of level of expression, both up or down regulation, results in atrial fibrillation. But what was not known until this work, we found that TBX5 has 3 different isoforms shown here. And one of those isoforms is sex dimorphic. So for example, you can see here in the healthy male and female atria, they're expressed at high level, slightly higher in male, but still both sexes have expression of this particular isoform, short isoform in the atria. However, if you look at failing heart atria, so now in the male heart, it's completely preserved expression activity of this particular isoform, but in the female heart, it's gone. It's completely gone. We didn't do mechanistic studies yet, but we plan to do it. And basically, this could explain one interesting observation that patients with heart failure very often develop atrial fibrillation. In some populations, up to 50% of heart failure patients develop atrial fibrillation. But what is known is that healthy women have lower propensity for atrial fibrillation, while failing women with heart failure have higher chances to develop atrial fibrillation. So perhaps knockdown, pathophysiological knockdown of this particular isoform of TBX5 is responsible for this sex dimorphism atrial fibrillation in patients with heart failure. Another example is CREM, which is a very interesting family of transcription factors. So this gene encodes close to 50 isoforms of transcription factors, which regulate basically parasympathetic response and also spermogenesis. Most of these isoforms are only expressed in sperm, but there are about 10 isoforms which are expressed in the heart. And they regulate, again, response to, beta-adrenergic and cholinergic response. So what we found, again, 3 isoforms shown here expressed in the heart. As you can see, based on levels of activity here, they are preferentially male isoforms. They are not actually expressed in female. And these particular transcription factors happen to be repressors. So they repress expression of very important genes involved in beta-adrenergic response, which perhaps, again, explains differences between male and female heart with respect to beta-adrenergic response. Again, interesting testable hypothesis, which we are working on right now. Next question we ask ourselves is, can we see any differences with age between men and women with respect to activity of promoters and enhancers? So again, as I mentioned before, we had a nice spread of ages between 26 and 72, I'm sorry. And indeed, when you compare differences in the atria, ventricles, how it differs with age, we found significant correlation for a number of genes, especially as you can see here, for ventricles. So simple correlation, Pearsall correlation, Spearman, correlation between age and activity of promoters and enhancers shows that in the male heart, it's basically absent. So it's close to 0, slight positive correlation. But female heart shows very strong correlation with age. And these are genes which are involved, which contribute to that correlation. So when we look more specifically at specific genes, we found that there are 1,800 genes which have the same trend. They are positively correlated with age in women. So they basically increase in activity of promoters with age, while there is negative or no correlation in male heart. And these are a couple of examples, all very important, of course, genes GATA4 and TBX20, as you can see examples here. In women, it's increasing in activity and expression of these promoters, while in male, it's actually decreasing. So another feature of this age-related activity is actually switch of isoforms. Not only total activity of a particular transcript is increased or decreased, but also there is actually switch of one isoform to another isoform. So we found that there is age-dependent promoter use in 1,100 genes. So it's a very significant, almost 10% of genome. And specifically shown here, there is, for example, 232 genes which switch isoforms with H in the male atria, 700 in female ventricles. But also there is about 100 genes which actually switch both in male and female, and there's no sex dimorphism. So again, some of these genes are listed here. If you're interested to look, I just give you a couple of examples. For example, here's the UCKL1. This is a female-specific downregulation with H. You can see here that there are two isoforms. I'm sorry, there's male and female in color, and you can see that with H in the atria is significant downregulation compared to male, and also in the ventricles even more significant. And there is also corresponding actually RNA sequencing data to that isoform. Another gene, CDH2, there is actually upregulation, but in both sexes, both male and female heart. So you can see here, male and female, there is upregulation of this isoform in the ventricles of this isoform, and there is also corresponding RNA sequencing transcript. HEN2 is another example, which is male-specific upregulation with H. You can see here, in women it's almost completely absent, and there is also RNA sequencing corresponding product. What is interesting about HEN2, I'd like to mention, in January, Circulation published an article, and Eric Olson wrote very nice editorial for it, showing that 4 transcription factors expressed in drug-inducible system in fibroblasts can reprogram the fibroblasts in the scar tissue into myocytes. It was a very interesting paper showing perhaps one pathway towards tissue regeneration. However, when I looked at the paper, I could not find any data about sex differences. So they just basically conducted everything in only male hearts, not female hearts. And you can see here that this could be a problem because HEN actually is a male-specific gene with the age, and probably was disease state as well. So perhaps this is a powerful transcription factor for reprogramming, but only in the male heart and not female heart. Again, interesting hypothesis to test in the future. So let me conclude. So basically, I showed to you that we constructed a comprehensive library of regulatory network in the heart, both healthy and failing heart. We found that actually ischemic and non-ischemic cardiomyopathy have significant differences. So ischemic is primarily metabolic disease, while non-ischemic cardiomyopathy is primarily immune, autoimmune disorder, as some now postulate in the cardiology field. We also found that SNPs, up to 30%, depending on the etiology, could be located within transcription start sites, promoters or enhancers. We also identified significant differences in regulatory network, both healthy and diseased hearts between men and women. There is switch of promoters and enhancers between men and women, but also change in level of expression. And also we found big differences between anatomy, disease state, and sex in general. So here are some specifics. You can read the conclusion. But basically, this is a starting point at which we would like to look at actual nuclear structure, nuclear architecture, how we can explain the switch of isoforms within the same gene, which takes place in literally up to 10% of genome. So it's clearly not a random phenomenon, and it would be interesting to develop new methods within physical genomics space, how to explain these observations. So I'd like to thank members of my lab, especially Anna Gams, whose work on sex difference was her doctoral work, and she defended her thesis this year. Eric Eritkin, Sharon George, Paloma Amaral, and Katie Trampel, who now continue this work. Thank you very much.